The next portion we're going to start is about the glia cells. They do not strictly glue, but their work is to support and bind the processes and generate the synapses. And they are 10 times more active as compared to the neuron or the nucleus itself. So there are different kind of glia cells. One is known as astroglia. They are star shaped, and they give physical support, produce scars tissues when there is an accident, the process whereby dead cells are engulfed and digested. This is what the astrocytes do. And they remove the excess neurotransmitters in the synaptic gap. Not much related to psychotherapy. Oligodendrocytes. So oligodendrocytes glia, their function is the production of the myelin sheath, and they are also known as the satellite cells. Then we have macroglia cells. They are very small, and they have no structure, and their work is to invade the alien cells, and they get activated when there is a tumor or infection in the brain. So when we go for the lab test, basically if there are more macroglia, it means that there is infection or tumor in the brain. Then there is Schwann cells. These peripheral nerve system cells help to support and insulate that system processes, give physical guidance to spooting or damaged central nervous system axons. So they give they help the axons to regenerate itself. They are also called the terminal Schwann cells, and there is some evidence that they are helpful, that they help in the regeneration. Let me give you another example. So basically what we have learned in the glia cells is that they assist and help the neurons and the axon in different functions. So how does the neuron communicate? They communicate with the action potential. And action potential is a must for sending the electrical impulses. Potential refers to source of electrical activity. So there is an electrical activity going in the neuron, which is known as action potential. The communication depends on the neuron's excitability. If the neuron is not excited, the action potential will not occur. Its capacity to react to a stimulus with an electrical discharge. So the nucleus activities which are going inside the neuron have to activate themselves in order to get the action potential. So action potential is produced by charged particles called ions, which are passed through the cell membrane. The extracellular and intracellular fluid both contains ions. So the ions are inside the membrane and outside the membrane. They are positively charged and they are negatively charged. So there is action potential occurs on the membrane, which is known as the electrostatic pressure. The force produced by this repulsion and attraction is called electrostatic pressure. There are many different ions unevenly distributed inside and outside the cell membrane. And in this distribution that gives the membrane an electrical potential is therefore called membrane potential. The membrane has an electrical charge because there are positive and negative ions, both inside and outside the cell membranes. The membrane is selectively permissible to ions that it allows only certain ions in and out. So there are different ions. The ions are sodium, the potassium, the calcium, and the chloride. The potassium, sodium, and chloride are found in the extracellular membrane, and the chloride is found in the intracellular side. The chloride and the sodium both are on the extra. No, I've mixed it. Perhaps the most significant ions are the sodium, the potassium, the calcium, and chloride. Potassium, sodium, chloride ions are found in the extracellular and intracellular fluid, although there are more potassium in intracellular fluid and more sodium and chloride in the cellular fluid. So chloride is more prominent extracellular ion. The type of channel opening and is governed by the neurotransmitter is called transmitter or ligate. The channels are called voltage gated, and it is these that they are responsible for producing the action potential. So the permissibility of the membrane, it is the ability to allow the potassium to enter, or it allows the potassium to exit, is dependent on how many channels are there. So basically there should be the sodium channel, the potassium channel, the calcium, potassium, sodium, and the chloride channel. So the distribution will depend on the channels and how they are functional. If inside, what is a membrane potential basically? If inside the membrane is negative, to the outside, positive ions will be attracted inside, so there will be the balance going on. So it keeps on working like this. As a result, negative ions will be forced out. The degree of attraction or repulsion is determined by the membrane potential. So if the membrane potential is appropriately working, the ions will be balanced in and out. The charge across this membrane is about 60 to 70 millivolts. When it receives no stimulation, this charge is called the resting potential. Because there are negative ions inside the cell, this resting potential has been attributably defined as negative 60 to 70. The potential is produced by membrane selective permissibility to ions and the different concentration of ions inside and outside the cells. The unequal distribution of the ions is maintained by pumps in the cell membrane. So when, for example, if the potassium is there, is less through the membrane, when the cell is at rest, thus the expulsion of the potassium results in the inside of the cell losing positive ions, producing a negative charge on the outside. Potassium does not leave the inside of the cell endlessly because at a certain point the membrane will force potassium to flow back into the cell. At 70 MV, the strength of the outward and inward flow of the potassium are similar. So this is how the balance is going to keep on working. So what is depolarization and hyperpolarization? When the sodium channels are opened, the cell becomes more permeable to sodium, and resting potential becomes more like equilibrium potential for the sodium. Increased permeability and influx of sodium constitutes what is called depolarization, and the positive ions make the membrane potential less negative. So as we continue, if this continues, the positive ions continue to flow into the cell, the intercellular charge reverses from the negative to positive. Eventually, the resting potential is reached, but it first overshot. When this happens, the membrane is described as hyperpolarized, and the process is known as hyperpolarization. The time taken from depolarization to polarization is about 2 to 3 milliseconds. So how the nerve impulse is transmitted. The reasons why the action potential does not die is that it has a reprolongated, recharged, and it makes its way to the axon. So as we knew that there were glia cells and other cells, they keep on repairing the axon and the dendrites, and the and the potential charge keeps on going. In unmyelinated axon, the impulse leaves the axon hillock where the depolarization occurs, where the action potential is triggered and goes through the repolarization. In myelinated axons, the impulse still contains for a short distance but is regenerated by the node of Rinvir. The nodes become depolarized and another action potential occurs. The potential occurs because voltage gate sodium channels are opened. The electrical message is thus passed along the axon by being regenerated at each node of Rinvir, where the passage of the action potential in unmyelinated is smooth. The course of action potential in the myelinated axon is slightly more jumpy because it is repropagated at each node. Therefore, there is a slightly delay before the impulse moves on because there is a jump. Because the membrane has open channels and allow the flow of the ions in and out. So basically, because of the channels, the information will become in a delayed form as compared to the others. So the next is the synapse. What is a synapse? Synapse is basically when an action potential is sent down the axon, depolarization occurs at the terminal button. This is not the end of the communication process. When depolarization occurs at the terminal button, calcium channels open, allowing the ions to enter the cell. The increased permeability of the cells and in presence of the cell is responsible for the secretion of the neurotransmitters from the vesicles. And the neurotransmitter is released into the synaptic gap by the process called exocytosis. This means the transmitter containing vesicle moves up the cell membrane of the presynaptic terminal button, pushes up against it and fuses in it. When this occurs, the vesicle releases the neurotransmitter, which moves into the extra fluid of the synaptic gap, where it binds to the postsynaptic of the receiving terminal button of the other neuron. So this is how the process keeps on going. The neurotransmitters is excitatory. It excites a cell into producing an action potential, and the results from the sodium and calcium ions going in and potassium ions being pushed out. This type of potential is called excitatory postsynaptic potential. Rest is the procedure, how it keeps on working. The decreased calcium is also found to accompany increases in the neurotransmitter release. When this occurs, the release of the neurotransmitter is said to be calcium independent. So basically, the calcium and the sodium works. How are they going to go in and how they're going to go out? What type of information is going to come? Which calcium is going to go into the synaptic cleft? What the membrane is going to allow, and how the dendrites of the other are going to get hold of the calcium or the potassium, and then the information is processed on.